I say that it's really pleasing to see so many people interested in astrobiology attending even on a sunny Friday afternoon at this time. So let's try to make this at least a lively discussion type of thing so that you don't need to fall asleep. But well, anyway, to start, so first I want to do some advertising here. I want you all to be aware of this event, which is the next EANA, European Astrobiology Network Association workshop, which runs annually and will be held next time in Turku in October. The actual meeting days are 22nd to 24th, but do take note that actually already on 21st we will have a public lecture in the Sunday evening. Topic will be first and second origin of life, and the first origin will be covered by Andre Prague, who is the chairman of EANA. Running out of pictures. And then the second origin will be covered by Sten Rasmussen, originally from Denmark, who is now working on systems for artificial life in Los Alamos. So I hope you all take note of this, and I hope you all would come if ever possible, because I really hope that we would make this like a joint effort to show the rest of Europe that we have astrobiology in Scandinavia. So we certainly would like to have all the neighbors there. So do take note, and do take note that the registration ends by end of August, so right after summer holidays. And for students, there are travel fellowships available from ESA, deadline by end of June. So don't miss application. You will get 500 euros free money. And then to the main topic, models for early life. We are losing the story. It comes there. Also, apology maybe. I have been on your list for seminar titles for quite a long time, with the title on life and climate. And this is because we were working a little bit on this last summer, and found quite exciting things, I thought. And there is actually a paper came out of it. But then I did find it quite too complicated, and I could not follow it very much further, so I have returned back to my favorite virology type of things. But anyway, if you like to take a look at the life and climate connections as we were kind of modeling them, so there is some nutrients on that work. But so being virologist, I am most keen on the early stages of life, molecular evolution, which probably may be something similar to viral life. So today I'm aiming then to describe you some of the very simple survival strategies of viruses, and viruses being the most simple life forms, kind of on the border of animate and inanimate things, like not really alive but not really lifeless. So we may think, of course without any proof, that they may be something similar to the most simple early stages of molecular evolution. So we try to aim towards that kind of story here. But to talk of simple life and early life, then we probably should first ask of what is life. So what are the basic properties of life, and what can we consider as living, alive? And of course, like first we have these images of very complex and large life forms, life consisting of trees and animals and anything moving around that we can see and observe. Although, of course, most of the life forms are consisted of things too small for naked eye. So invisible microbes, being either eukaryote, algae, or fungi, or whatever, either unicellular or multicellular, like algae like that, or small animals like that, or then just single-cell fungi, or then even more simple life forms being the prokaryotes, bacteria and archaea, which in a number of species and also in biomass probably apparently exceed the mass and number of any eukaryotes on Earth. So different life forms being very variable, from very simple to very complex, and somehow we should then make some kind of conclusion from this whole variability, that what life is all about. And actually, any definitions of life are rather difficult, or probably almost impossible to give, also for the reason that we know here only is one example of life. So we know one representative, and we can describe what this life is like. We can give properties of life, but we cannot really define life in simple terms. But here I have a list of some of the most central features of life, some of the things what we can consider like hallmarks of life. For instance, one of the basic features is that life needs to be reproducing to provide its own maintenance through time, so that it is not degraded over time, but can maintain itself by self-reproduction, and also being separated from the environment by being confined in cells, in cell membranes. Also within the cellular structure, we can be very impressed of the very high complexity of life. One of the hallmarks of life has been, for the physicists, I think it has been the very low degree of entropy, meaning, in other words, very high degree of order and organization, very complex molecules and structures and functions, mediated by very complex biochemical reactions. In aqueous solution, we need the water environment, and for all these complex reactions, we need enzymatic protein enzymes to catalyze these reactions. Most of the biotic reactions are such which would not happen very spontaneously, at least in biological conditions. So we do need enzymatic kind of enforcement or guidance to enforce the biological reactions, and also to have them happen correctly and in coordinated, regulated fashion. So life has to be very orderly, coordinated. It's not chaos, it's order. Okay? Okay. Also, then these are kind of very complex features. Although they are hallmarks of life, but they apparently have not been there from the very beginning, and we can kind of also wonder at what stage they were acquired, and whether all these things had to be there before we can call it life. So kind of what is the point of transition from inanimate to animate. Okay? And the way how many of the features have come has been by interactions with the environment, by adapting to environmental conditions to drive evolution and then produce the suitable functions now which are encoded in the genetic.
Can I ask you a question?
Sure.
I mean, is it understood why it has to be an aqueous solution? In other words, why would it have to be water? Couldn't it be in some other?
Okay. Well, firstly, we need to have the ingredients being imported to the reaction sites and then exported, like waste products and products transported from one side of synthesis to the side of utilization. There has to be a lot of exchange, and because it's exchange of molecules, it has to happen within a carrier.
That I understand. Yes, I was just wondering why not ethanol instead of...
Okay, ethanol would be completely volatile or evaporating, at least in these temperatures, and liquid form only in quite low temperatures, which would make reactions then again quite slow. I think ammoniac might be a suitable solvent, but again, liquid only around -70, around that region, and in very narrow area. And water has very superb properties for being a liquid carrier, long range of liquid form over the large temperature, and good solvent, and hydrogen bonding between molecules, and just the best. Some others might work, but water is really the most optimal. Okay, and so the genetic information was the essential carrier of the information that makes things happen in a coordinated fashion. This is probably very familiar, at least to most of you. So coded in the DNA and then expressed via transcription into RNA, which is very similar to DNA, but the essential difference being that the RNA is a very fragile molecule. Like DNA is a double helix polymer, long strand of nucleotides. RNA is a single-stranded polymer composed of very similar nucleotides, with the exception that there is one OH group in the sugar backbone in each of the nucleotides, which makes it very reactive at each side of the OH group. And that makes RNA strand chemically very reactive, which again makes it very unstable. So it is a good molecule to carry the information in a very transient way from the DNA into the processing. And the processing is happening so that the nucleotide sequence carried by the RNA is translated into amino acid sequence to make proteins. And then the translation is happening here in a so-called translation machinery. There you need the message, the RNA sequence, and you need ribosomes. The central parts of the ribosomes are also composed of RNAs. And then you need transfer RNA molecules. So these are also composed of RNA that carry amino acids into the reaction. And here in the ribosome, then the amino acids are fused in the sequence determined by the genetic code into an amino acid chain, which then eventually falls into very specific secondary structures to make the functional proteins that are then the really essential working tools for all reactions and building of structures. Okay, but the system here contains this very classical chicken and egg problem, which is that a whole lot of different proteins are required in each of the steps. First, in maintaining the genetic information in the DNA, in the synthesis of DNA, in stabilization of DNA, then transcription of the RNA, processing of RNA, and then also here in the translation stages. So this seems to be very much like a very cyclic process, like the chicken and the egg. And so it has been kind of confusing to know where it did start, or if you had first the tools or the information to make the tools. Okay? But then now, a few decades ago, it has been pretty clear that to get the whole system running, you first have had to have the machinery for making the proteins. And so, as I was mentioning already, the machinery, pretty much the basic parts are composed of RNA. So it seemed that the RNA had to be there first, and also the RNA then could contain the first codes for translation of the proteins. So this is kind of a quick explanation or proof for the hypothesis that the RNA-based life had to precede the life what we know now. Like the RNA life had to be there before the DNA-based life. And also that the protein synthesis was started based on the RNA, but apparently the RNA had to be there even prior to the protein synthesis. Okay, and now as I said that I'm aiming to compare these two viruses, then a lot of the functions and features of the RNA protein world, hypothetic world, would be fairly similar to the viral functions as they still exist today. So now I would like to go through some of the hypothetic steps of how we think now that the RNA world and RNA protein world came about, and also point out some essential problems. Like this is certainly, we think that we know the basic steps, but it is certainly not trivial. There's still a lot of open questions. I wonder how I'm doing with the time. But anyway, so to get anything going, or at least to get RNA started, you need the building blocks. That means you need the nucleotides. For nucleotides, you need the components of those, which would be the bases and the ribose. So the four bases, purines, adenine, guanine, pyrimidines, cytosine, thymine, and that is actually for DNA, and this is for RNA. And the backbone component for RNA is the ribose sugar. And now chemists have shown pretty convincingly, as far as I can tell, that the bases can be actually synthesized by prebiotic reactions. Now, I mentioned earlier that for just about all of the biotic reactions, we need enzymatic catalysis. So the cells can make all these things quite easily with enzymes. But making them without enzymes is much more difficult. They hardly can form in prebiotic or non-enzymatic conditions. So the enzymes would be the tools for making them, but without enzymes it can happen. And particularly it has been shown that the purines can be synthesized from hydrogen cyanide, and those can be synthesized from formaldehyde, acetylene, and urea. But it is certainly not that easy as we say, you need really high concentrations of the precursors, and apparently conditions need to be really highly optimized. It seems to go best in really cold conditions. Electric ices, that would be like minus 18-degree ice solutions. So very cold conditions which help to condensate and stop the molecular motions. So it is not trivial. You also need some UV radiation for isomerization of the condensing polymers of hydrogen cyanide to make conformational switches, then to eventually make it turn into a heterocycle like that. Okay, but even that it may be difficult, it is feasible to make the nuclear bases. While making the ribose is apparently, at least as far as the chemists know now, making of ribose is much more of a bottleneck.
Could you make those in space? Ices and grains in space?
Now, as far as I remember, they occur in Murkinson meteorites.
Mm-hmm. But no ribose, of course.
No ribose, no. But the nuclear bases occur. And several isotopes or analogues of nuclear bases. So actually a problem with these reactions are also that not only these heterocycles are made, but a whole variety of very similar analogues. So one big question is, how did life end up using just these? Because the same non-controlled reactions are making a lot of different analogues, as well as for the bases, but also for sugars. And making of the ribose is really difficult, particularly because of the reason that the hormone reaction, which is producing sugars from these starting reactions, is making a lot of different sugars, and they are very easily kind of converting from one to other. So you cannot really accumulate ribose. And then if you do accumulate ribose, then the more difficult step is combining the bases to ribose to get the nucleoside. So I think this is probably, maybe some of you know this more better, but the combining of the bases to ribose is the most difficult step at the current knowledge.
Does one know why life is using phosphorus?
Yes.
Because it's essentially not abundant as well, whereas all the other atoms and molecules they're very abundant in the medium, whereas phosphorus is essentially
and this is one of the obvious bottlenecks for this whole cascade, is the phosphorylation, which is the next thing that is required. And particularly you would have needed organophosphate for the phosphorylation, or phosphate molecules for the phosphorylation step. And that apparently has not been soluble, or at least available in the oceans at the early time. Although who knows the conditions of the early time, maybe the geologists know this better. Although it has been postulated that there may have been apatite dissolving slightly in the oceans. So a little bit of apatite available, and that can be made to react with the nucleosides with very strong heating in urea, ammonium chloride, but so not easy business. So we don't know. But anyway, this is what should have come out of those the starting ingredients. So nucleotides, and particularly of this very kind of nucleotides. So ribose, sugar combined to purine or pyrimidine bases, and then combined by a phosphate ester linkage. But then, as I have been pointing out here, is that major problems in making this kind of compounds would be that even if we had the right conditions, they would produce a lot of different things also. So, in addition to adenine and guanine, at least these and maybe many other purines, multiple different pyrimidines, and the sugars, as I said, they easily interconvert. Like if there's 5 carbon sugars, they have isomers, which just have the hydroxyl group in different positions. They spontaneously change from one to other. And the ring, so we need the ring molecule, and we want the furanose, which means that it is circularized from the carbon number 4, from there, to carbon number 1. While just as well it could be a furanose, which makes a 5 carbon ring. So needs very specific chemistry to make that. And also could be 4 or 6 carbon sugars also. And then we have this stereochemistry, which also has to be right. So the ribose ring has to be in correct conformation, both of the one to the base has to be in correct position in carbon number 1, meaning that it has to be, in this projection, it has to be pointing up and not pointing down, which would be the alpha form. And likewise, the orientation has to be correct here, so that the carbon number 5 is pointing up, meaning it's the T form, while the L form would be completely non-functional. And again, now if we talk of prebiotic synthesis reactions, then all different isoforms are made in equal amounts. So we don't really understand where the specific chiral forms or how they became selected. Also, the phosphate bonds could be oriented in many different ways between these nucleotides. So they have to be going from 3' carbon from here to the 5' carbon here, and not something else. There's a lot of different alternatives. So it has to go right, and we don't understand at this point how it happened to make just these things. But anyway, so this is a little bit of the same thing again. So we had to get the correct nucleotides. They also have to be phosphorylated at the 5' carbon. This means that with the 3 phosphates, they had adequate chemical energy charge here, which then allows the bonding between different nucleotides. And this is how it happens today. It needs enzymatic catalysis, but if the phosphates were not available, then how were they charged? So that's one question. And without enzymatic polymerization, it seems that even higher energetic charging would be required. Nowadays, for instance, in laboratories, people are using aminopyridines to activate this phosphorus group, so that then the nucleotides can kind of somehow gradually polymerize. But if we think that now we can find this kind of aminopyridines for activating molecules, it probably was not available at that time. These aminopyridines are analogs of bases again. So somehow there were very complex molecules available in the early sides of life which make this possible. Okay, but anyway, somehow the polymers were assembled. Somehow, and now even in the laboratory conditions, people can achieve spontaneous assembly of up to above 50 nucleotides in spontaneous non-enzymatic polymerization reaction. But
So that's always template directed, right?
no, it would be just like oligomerization of nucleotides.
Without a template.
Without template. So oligomers of up to, so in the last thing what I heard in a meeting 2 years ago now, actually, so it may be more now, but the last thing what I have heard was about 50 now. And 50 might be enough to get some kind of functional molecule. And this kind of a polymer assembly is very much aided or enhanced by having it happen on clay surfaces which stabilize the polymer, kind of prevent the degradation and hold it together. So particularly montmorillonite clays are very good for stabilizing assembly of this kind of polymerization. So somehow the polymers were assembled. But then in order to get anything lifelike started, then something had happened to make them replicate. Now we are finally coming to the first stage of life. So apparently the replication was the first hallmark or the first requirement to start anything to happen towards life. But now, at least any of you who are working in biology would know that there are many different problems in non-enzymatic polymerization. First, to get the nucleotide bonding take place is one problem, but then another one would be that if you get a double stranded RNA constructed, it tends to be a very closed structure. The next problem is getting them apart again so that you can then replicate them again. So these things are done by enzymes nowadays, but it is a very big question on how it happened in the early world. This separation of strands might have happened in very alternating conditions, like cyclic changing, for instance, of temperatures or salt concentrations, something what kind of made it anneal and then made it come apart again. So you needed some physical forces to do the work what enzymes are doing nowadays. Okay, but no enzymes. But it seems that there had to be something helping the reactions. And now we do know that some of the RNA sequences can have and do have catalytic activities. So maybe the RNAs were actually quite early on taking catalytic functions. And we know nowadays, for instance, that RNAs can do several different types of functions. For instance, join amino acids together to make peptides. They can amino acylate the RNAs. So these would be needed for the protein synthesis. But for making of the nucleotide polymers or RNA polymers, you might need this kind of reactions, which would be like cutting and ligating RNA itself. So a lot of RNA sequences, some RNA sequences are able of cleaving the sequence itself and then again ligating it. This is called splicing also. So this is known to happen still in many different RNAs in the cells. For instance, in the messenger RNAs, they can cut and ligate themselves. The tRNAs are still processed by RNA enzymes to make them mature. Likewise, as are the ribosomal RNAs. So RNA cutting and ligating can be done by RNA. But polymerization is not actually a feature of RNA.
Can you maybe briefly say what is the significance of both cleavage and ligation? Why is it important?
Well, we know that RNA can do it.
Yeah.
So at least it is something that can be achieved. And if you then think that it is able to cut a strand and paste it together, it means that it may be able to reassemble things or build up things. Here are just first, I come back to that. But some pictures of this self-ligating or cleaving and ligating ribozyme sequences. So we are calling these RNA enzymes or ribozymes. And these cleaving and ligating reactions are some of the most typical. And actually you don't need much of an RNA sequence to make such an active structure. The smallest natural ribozymes, 49 nucleotides. So we are coming close to the 50 what people are now able to polymerize. So these are some viroids, plant pathogenic viroids, small circular RNAs that make this kind of so-called hammerhead structure. And the structure is essential in the terms that when it's folding like this, it is holding these reactive nucleotides in such position that they can do a transcriptification reaction in a specific position. So actually the very reactive OH group in the RNA ribozyme can interfere with the phosphate diester bond and break it away. So actually the reactive property of RNA, although it makes RNA very fragile, it also makes it reactive in the sense that then it can drive chemical reactions. So it is food and path. Okay? And again, here is another example of a ribozyme sequence. This is a little bit more complex. This is just showing that this kind of a ligation reaction can be made to evolve in vitro, in test tube. People can just kind of drive it and select it so many times that it becomes a polymerase which actually adds single nucleotides. But the polymerase activity remains very low. So the ligation activity is much better than the polymerase activity. And now just to point out that if we consider this as some kind of enzymes that make the reaction happen, so it is kind of based on the structure of the RNA. So we can think that it is an informative sequence. It's not going for any proteins, but it is maintaining the structure, and the structure kind of then is the functional part. The sequence kind of produces the functional structure.
So roughly how many different ribozymes are known?
Dozens.
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 Not many more than that. And the catalytic functionality really only works because of its folding properties, isn't that right?
Yeah.
Yeah. And that folding is accomplished, as you say, by the reaction between the OH on the ribose and the phosphorus.
Yeah. So OH in position 2 in the
ribose. There's no base pairing at all.
Not necessarily.
In that one, yeah?
No. And I also here want to still point out the significance of the structure. So in this very simple ribozyme, we only need to have these nucleotides conserved, while the other ones can be anything as long as the structure is maintained. Right? You can have different nucleotides here as long as they base pair. So the structure needs to be maintained. Okay, so RNA structure can do a lot of things. It can do some things, not a lot of things, but some things. Okay. So they could do some things, but they still would have a lot of problems. They had to get all their building blocks from the surrounding. It is still very problematic of how they were able to assemble enough nucleotides to long enough polymers. The original homochirality is a big question, and then obviously to become alive they would need to be compartmentalized at some point. And the replication by the ribozymes would have been still very inefficient. RNA are very fragile, very unstable, so it is a big question of how they would reach adequate size to become functional. And then it's a big question on how these particular components were selected, how these 4 nucleotides and how it was with ribose sugar and the specific chiralities.
Can I ask, you mentioned homochirality as a problem. I mean, in what way is it a problem? Is it a problem because it's there, or?
Well, the origin or how it was achieved, because the prebiotic reactions would produce both chiralities in equal amounts, and still only you can build polymers only with homochiral nucleotides. So somehow you had to be able to select just the one chirality.
But do we know that the selection took place already in the beginning? Isn't it possible that it came later?
At least you would not be able to build the polymers from mixed chiralities. Because they would become kinked. They would not be smooth and straight.
At least not very long ones, and actually extremely short ones only just 1, 2, 3. We would do it by random, in a racemic mixture.
Okay. Then to get any protein synthesis started, then amino acids were needed. Now we are using 20. Maybe early on it was just maybe 8 amino acids were used in the protein synthesis. And as Stanley Miller showed already in 50s, they can be fairly easily synthesized by energy charges from hydrogen cyanide, ammonia, aldehydes. But again here with amino acids there is the chirality problem. To build polypeptides, amino acids need to be homochiral again. And the chirality here is built in the 4-way symmetry of the central carbon molecule. Now, because all the side chains are different, and they can be pointing out either one way or in a mirror image way, as shown here for the alanine molecule. Axel has probably explained this many times to you, so it's familiar thing.
So a very naive question. Are only the L forms produced in the cell, or, I mean, we know they are used, but do they produce both types of amino acids in the cell and then dismantle the stuff that don't need those?
Some bacteria at least synthesize also the D-chiral amino acids and use them for some cell wall components, for instance, for material which is not easily biodegradable. So they can utilize it for stable structures. But apparently now again the enzymatic reactions are very specific and they make only the correct things.
But there are quite many D forms of amino acids in gram-negative bacteria, and aren't there more around surviving? I mean, I don't hear that homochirality came up early. I thought it came much later.
Now, again, I think that the functional polypeptides would require a homochiral.
In the RNA world, yes.
Yes, okay, yes. Yeah. Now, I don't know if how the peptides would function in a mixed chirality.
I mean, would you assemble mixed ones? I don't think so.
No, I don't think so, but I think there are surviving D forms of
as monomers. Dimers perhaps is the most.
How is it in microbes when they make the D forms? Do they polymerize?
I'll have to find out. I've just seen somewhere in the books that they I don't know if they're functional in peptides and proteins. Maybe they are not. I mean, I have to find out first.
Yeah. At least you would not make functional proteins from them, but any.
So they may appear in other types of amino acids or even.
So
they still do appear in life.
They do.
We have them around.
Yeah, and they can be very toxic also.
Oh yeah, sure.
Yeah. But anyway, I just want to quickly flash here this hypothesis from Anthony Poole and co-workers originally. I suppose this is still quite valid. I have liked this very much. I think this was one of the kind of the key enlightenment experiences for me in past years for the origin of translation, because this really makes sense. So that translation somehow at some point became initiated kind of accidentally by a machinery that did exist already for RNA replication. And apparently the early components of translation machinery were there already, like the RNAs and the ribosomes. But I suppose you have heard a very detailed explanation of this earlier.
Not yet.
And Alan Folger did it much better. But anyway, so the translation probably certainly not proven, but I think this is a nice hypothesis that the RNA replication had to exist before protein synthesis started. So there was a molecular machinery for the RNA replication, and so if these are the oldest RNA components that we have in the biosphere, so maybe they were replicating RNA first, and then they were kind of maybe accidentally initiating the synthesis of forming of the peptide bonds and the synthesis of polypeptides. And then when protein synthesis evolved and proteinaceous enzymes became available for replication, then this machinery was totally left for the translation, and replication was then done by the protein enzymes. And a very important step for the evolution of life. Certainly we would only have some short RNA molecules hanging around in the world if this step did not happen. We would not have any cellular life. So that step started the so-called RNA world or RNA protein world. So RNA genomes encoding for protein products, which initially, certainly, and maybe for quite a long time, the protein products were still quite random and very simple and not mediating any very sophisticated or very accurate functions. But maybe they were still very useful for stabilizing the RNA. You know, like I mentioned now several times, that the problem of the RNA is that it is so fragile and it breaks up before it can be even synthesized. So binding with proteins would make it very much more stable, and also the proteins would stabilize these catalytic RNA structures so that they would become more stable and efficient.
So can I'm thinking about this one statement that you made in at the end of the previous slide about this conversion from a translationary machinery to a replicating machinery. You said that was critical for cellular organisms, and I'm very surprised about this. I mean, I thought all of this would have required already the presence of some kind of a container or a cell?
It might have required a container, but that is a separate question. It's possible that there have been some inorganic containment. It is actually difficult to think that these reactions could have happened in open waters, for instance. So maybe containment was there, but certainly without protein synthesis you would not have had a capacity for making proteins for transport across membranes, or even for lipid biosynthesis. So you would need the protein components for making cells, even lipid membrane envelope cells. Okay, but anyway, so not efficient biosynthetic pathways for precursors, maybe from surroundings, and with very primitive ribozyme enzymes, and maybe even with the very early protein enzymes, replication is very inaccurate, making a lot of mistakes. And now these organisms have to somehow prevent accumulating too many mistakes. And the longer the genomes, the more mistakes you accumulate. And this is kind of like a statistical inevitable condition, that you have to maintain the genome short enough so that the probability of getting mistakes is not too high. So short genomes for avoiding mistakes, or for avoiding accumulating mistakes, and also short genomes because the long ones would first have been too fragile to maintain. So two reasons for the thing that genomes remain very small as long as they were in RNA form. That means very small coding capacity, very few gene products. So how to mediate functions, how to regulate functions. Okay, RNA viruses still today are very much in the same or similar situation. And in many ways they probably resemble those early RNA protein organisms. So genomes are RNA based, function directly as translational templates. Genomes, for the same reasons, have to remain very small, coding capacity only for maybe 3 genes, or up to maybe 14 genes. So this is the variation what different RNA viruses contain. Smallest ones, 3 gene products, and the largest ones, 14. Fully heterotrophic, so no means for synthesizing their own building blocks, but they have to get their nucleotides and amino acids from the host cells. Also need to utilize the energy products and translation machineries from the host cell. So they do not have cell membranes. But as you were saying, some kind of enclosure is really necessary. And now, of course, viruses are enclosed within the host cell. So maybe again, analogous to the early organisms being enclosed in something, some sort of inanimate cells or something precipitates. And only some gene functions are encoded, so they have to be something really essential. And typically viruses code for a replicase, and it often comes in 2 different enzymatic functions. One is a helicase, which opens up the duplicated strand, the double-stranded structure, and one is the actual replicase. So we need actually 2 functions for replication. And then one essential product for viruses is a code protein. So they need some kind of proteinase assembly to protect the RNA. So code protein and replicase. And then nowadays they also need at least one gene product to cope with the host. For instance, to separate the host defense or to kind of tame the host. So it seems that viruses are able to tame their host cells quite effectively. Then we of course have a special case of viroids. Now these are very small circular RNA molecules which are very pathogenic in plants, 400 nucleotides, and do not code for any protein products. Okay? And now, when the viral genomes are so very small, so they have to utilize extremely simple structures for all their functions, but they can do them very efficiently. They can replicate it efficiently, they can very accurately initiate replication. There are two different methods. They can initiate translation very efficiently, and they can maintain the RNA very efficiently. Also, viruses may resemble the early organisms by the feature of recombination. It's quite clear that viruses have kind of evolved to the form what they are now through massive recombination. They are just kind of doing this horizontal gene transfer in a kind of like completely wild way. Okay, and they also support replication of parasites, which probably the early organisms also did. I think I just quickly run through pictures. If you want me to go slow or stop somewhere, then we do, but otherwise I just show you. This one very efficient replication system for these very smallest viruses, like the viroids, the satellite RNA viruses, and also some small DNA viruses, is this rolling circle mechanism, which does not actually need any kind of accurate initiation, because it is just going around a circle, so the circle is initiated here, and long concatenation can be produced, and then there is a mechanism, and it is now actually this ribozyme cleaving mechanism shown there, which can accurately cleave the products into from the right size to make them the unit length products again. So the only accurate thing what they need is the site for the cleavage, and that was formed by the specific sequence as I showed you before.
So you do the cleavage, and then is that necessary because otherwise you cannot translate a long strand, only shorter ones, or why do you need to do the cleavage here?
Now the cleavage is because the replication here is actually a continuous process that is producing multiple rounds of the same circle. So it is going into one long thing, and then you need to cleave the long thing into unit lengths to make it the same thing again. And it can go either through this kind of linear form, or then it goes through two cyclic forms. So very little sequence information is needed, but it is very still very accurate. Another way for replication initiation is this tRNA-like structures in the end of virus genomes, and now this would be linear viral genomes. I told you earlier that these tRNAs are probably the very oldest molecular structures in the living world, together with the ribosomal RNAs. It has been proposed that they have been maybe some kind of replication initiation sites in the very earliest world. And now it is very, very clear that many of the RNA viruses are still using the same kind of structures in their ends for this replication initiation. So they seem to be kind of, probably they are relics from the old times. So that somehow the replication machinery recognizes this structure and can start the replication from the very end of it.
This is not this roll-up thing anymore, or is it?
What?
This is not this roll-up mechanism anymore?
No,
Yes, it is.
this is for linear
Yes.
RNAs. So the roll-up mechanism was maybe the simplest one, and this is a little bit more complicated, but this is at least as old as any linear RNA replication. So viruses still use it. And many viruses have such terminal structures. They vary in the accurate position of loops and in the accurate sequence. They can be quite different, but they are functionally similar because they all accept amino acids, so they function like tRNA structures. And they are very highly conserved in related viruses. So that if you mutate, these are viruses with 3 genomic components. So if you go and mutate, for instance, the RNA3 end, so it is very quickly converted back to the same old structure. So viruses can repair, fix any problems in what are introduced into the genomes. They can prepare them by just recombination between homologous sequences, or then by just very efficient selection by the kind of the hypercycle mechanism as described by Eigen originally. Viruses have quite varied structures at the 3' ends. This is again the TMV what I had in the previous picture also. So there is the tRNA-like structure in the end. And then they often have other structures in the 3' regions, non-translated regions. And these often function as very efficient translational enhancers. And this means that the 3' end, this means the far end of the RNA, then reacts with the first end, or the front end of the RNA, and make them circularize, interact with each other, so that structure then enhances translation initiation. Some viruses utilize proteins to do this translational enhancement, so that proteins are needed to bind in these pseudonodes, and then the proteins make the connection to the other end of the RNA. There are many different structures in different viruses. That was satellite TMV RNA, for instance, that kind of long structure, and this is alfalfa mosaic virus 3' end with multiple loops, and each one of the structures functions so that they find this one finds like the viral code protein, and these ones find some translation initiation factors, and then they mediate the interaction between the two ends of the RNA. And
Sorry, may I ask you?
yeah.
When you draw these two-dimensional pictures,
Yeah.
how important is the third dimension? This seems to be to me neglected.
Yeah, right. Okay. Projections are many different what you can draw out of these, and depending on your computing system, they can even look quite different. And I'm not an expert in the visual projection thing. But the main thing is to find the stems and the loops, and then find also how the loops are connected.
This loop could be sitting next to another one.
Yeah. What did I have here earlier? Something really. They just get very complicated when you try to put it into 3 dimension.
Sure.
So this looks more understandable. Yeah, but you're quite right that in real life they are really kind of intertwined into a 3-dimensional ball-like things. But even if it's like a 3-dimensional ball-like thing, the connections between the specific stems and loops can be very accurate, which is pretty amazing. For instance, now in here. So in the previous one I was showing structures of viruses where the 3' and 5' ends are interacting through protein connections. But there are also a lot of the smallest RNA viruses have the 3' and 5' ends interacting directly by kissing RNA loops. So now you need to think that this loop here would be in the 5' end of the RNA, and this loop structure here actually starting from the 3' end there. So this loop structure is in the far end of the RNA. So the RNA is what, 5,000 nucleotides long, and one end is here and one end is here, but by these very specific loops they are connected. And if you go and destroy these few nucleotides, the virus dies, like it cannot translate anymore. And these are found now in many different viruses, so it's nothing very rare. But very few nucleotides can make a big difference. And as you can see, the whole structure takes maybe just 20 or 50 nucleotides to make the stem and loop. And it is only maybe 5 or 10, something like that, nucleotides that actually interact. So it is a very minimal amount of information which is actually doing a lot of work. So I was earlier saying that we need a lot of RNA to make functional strands. But then when you make a functional strand, the function is in a very small size. So I think it is pretty neat. And like there are examples of these connections in quite many viruses. Also, I think this raises a question to me at least. Like if you think that the earliest RNAs have big linear strands, like they have big linear polymers. But then quite apparently, fairly early on, before the appearance of the real cells, the circular DNA genomes appeared. I think it is now thought that the Luca was a prokaryote-like organism or what, with circular genomes. So somehow the linear genomes had to be circularized. So I think it's quite apparent that it has been going on through this kind of base pairing between the two ends and functional connections between the two ends. Now this is complicated. Maybe I just jump this. But just say that the interaction between the two ends is also important because translation of the genome starts from this end and replication starts from this end. And when one is going this way and the other one is coming this way, they will collide. So there has to be mechanisms to prevent one or the other. And this one, for instance, is such that the replication when it starts, so this is a replication, it starts from the 3' and it goes along. So the replication is actually opening these base pairing connections between the 3' end and the 5' end. And so now when the base pairing connections are opened, there is no more translation, and now it is available and free for replication.
This is RNA, not DNA.
This is RNA.
Yeah.
This is small viral genome. There can be also different ways for switching between translation and replication. You know, like I have been telling you that the 3' end effectively regulates both replication and translation. And some viruses, for instance, can have the RNA folding in two different kinds of conformation, which are mutually exclusive. And this is, for instance, for alfalfa mosaic virus. And in the presence of code protein, it folds like this, and this stimulates translation. But in some other conditions, it folds like this, and this stimulates replication. Like RNA has probably a really wide alternative of different configurations. Like it can make base pairing, internal base pairing between many different sites. And these would be like two different alternative configurations. So it can trickle. So somehow like one linear sequence can fold itself this way and do one thing, and fold itself in another way and do another thing. And then here we have just a little reminder of this recombination potential. So we have a lot of viruses which quite clearly are recombinants of different things. This, for instance, is a so-called delta hepatitis virus occurring in humans. Hepatitis, I suppose so. But one part is a viroid-like part, which I earlier told you are the very small non-coding circular RNAs. And then the other part is quite clearly a messenger RNA recombined from eukaryote cell. And a lot of viruses can be kind of divided, well, they are divided into genes, and we can see that the different genes are each coming from different sources. So it is now said that they are undergoing some sort of modular evolution, so that each of the genes is kind of evolving on its own, and then they are just kind of combining to make functional combinations that is a virus. But like the same genes can be utilized in different viruses. Okay, means for stabilization. This is the viroids again. These have a very special means to make themselves stable. So this is the circular RNA. But these bind internally into this kind of very dense internal hybrid. To make it kind of like a rod-like structure. And even that RNA by itself is usually very fragile. You just look at it and it goes away. But this is so stable that you can autoclave it, and it doesn't mind at all. So you can actually test for viroids so that you autoclave your stuff, and then you see if it's still infectious.
And autoclave means up to 400 centigrade or more?
120 and increased pressure. Okay. Another way how viruses can stabilize themselves is that they induce formation of membranous vesicles, and they make the replication happen inside these vesicles. And they have been pictured for decades in like very nice micrographs. And this mechanism has been very nicely worked out by Paul Alquist's group and others also, showing that the viral replicase proteins actually associate with the membrane in such a way that they kind of cover the membrane surface, and then they cause a curvature of the membrane, and by interacting with the different viral RNAs and protein components, they can kind of withdraw all the necessary components inside the vesicle, and now they have a own little niche, or kind of like a protected replication environment, which is making the RNA stable against degradation, and also concentrate all components. So these might be kind of like miniature cells, maybe something which might have led towards the evolution of real membranous cells. This is second to last. Thank you for your patience. Anyway, so viruses are doing a lot of different tricks with very little of genomic information, very little nucleotides. And then we can even visualize or vision or think of pathways from the viral race or viral world into or towards the cellular life and DNA-based genomes. For instance, it is quite clear that there is a whole group or different groups, different genuses of viruses that can convert RNA into DNA. That is a part of the life cycle. So RNA can be made into DNA. Then we also know that virus-like elements, particularly these retroviruses or retroelements, can integrate into genomes. They can also transduce, carry different pieces of DNA with them, so they are very efficient vectors for horizontal gene transfer. They have carried with them a lot of different genes from one species to the other, and also a lot of eukaryotic genomes. Some of the plant genomes, the very large plant genomes, are almost completely composed of retroelements. Barley is nearly 90% composed of retroelements. So the genomes, eukaryotic genomes, can be mostly viral. And then also, if we think that the original replicating RNAs were maybe virus-like, and then they were somehow converted into circular functional genomes, so maybe the linear RNA virus-like genomes were the original components of any of the DNA genomes. So the original life might have been in the viral world. But it has been also a question of whether the eukaryotic genomes, these linear large genomes, would be more similar to the original linear RNA genomes than other the circular prokaryotic genomes, meaning that the original eukaryotic genomes is actually a quite big open question. And why do the eukaryotic genomes structurally still resemble so much these linear virus-like or RNP-like genomes? So it is quite possible that this kind of viral interference may have been involved in the initiation or invention of the linear eukaryotic genomes. Anyway, this is a long story. I appreciate that you have all stayed here. Thank you very much. But anyway.
Thanks, KLehto, for alluding us to this very interesting story. Questions, please. Yes?
I mean, when you show, for example, how they had worked out this mechanism of how RNA was protected.
In the vessels.
I mean, being totally ignorant in this field, how do you work out something like that? I mean, is this done with molecular biology techniques, or can you have some instruments where you can monitor this process?
Did you mean this?
Yeah, if you move back a few slides, I can show you. Yeah, no, next one. There. To the right, you said that there was a group who had worked out this mechanism, which seemed to me rather complicated. I was just wondering in practice, how do you...
This is very much based on electron microscopy, where these membrane vesicles can be visualized quite nicely, actually, and also the proteins can be localized inside the membranes by immunolabeling, and also these different protein-protein interactions can be also separately be tested in test tube, as well as the RNA protein interactions. So you can test the interactions, each one on its own, but you can then also visualize the whole thing.
Okay, I see. So it's really electron microscopy, which is
yeah, for this part it is. Yeah.
Other questions? The melting of RNA. What is the time scale? I mean, first of all, I mean, melting is familiar from DNA. Is such a thing actually happening with RNA as well? I mean, for the replication.
For the double-stranded structure, yes.
So double-stranded RNA.
Yes, any double-stranded structure on its own is quite stable. You know, like it is held together even by the hydrophobic interaction of the.
So the typical time scale, or maybe the shortest time scale on which you can still do the melting and also the recombination again, how short can that be? Are there serious restrictions?
Now,
because I mean, for PCR reactions, for example, you normally talk about half an hour and stuff like this. I'm wondering, can you do this actually on a time scale of maybe seconds or minutes?
Yeah. So the enzymatic reaction goes really fast. In a PCR reaction, it goes. Kind of a standard rule would be like 1 minute 4,000 bases, but in reality it goes much faster. Would anybody know the replication rate number? Yeah. It's fast, but I cannot know.
So it can be on the order of seconds, or?
Yes.
Yes, I see. I mean, one of the reasons I'm asking this is, of course, because there are ideas of doing this cycle, of course, within a single convection cell, which you may have in any porous medium, like, for example, in hydrothermal vents, for example. These hydrothermal vents are believed to be very porous, and any of these little pores can constitute such an environment where you can replicate in connection with such a laminar convection.
And melting by physical kind of a temperature, for instance, temperature shift would be slow compared to the enzymatic reaction.
Right. Any other questions? Yes.
So viruses nowadays, need DNA-based life in order to.
They need the host cell. Yeah.
So why this doesn't then sound like a good model for early life in a way, because how would they then function when those were not available?
Right. Okay. And this question is raised quite a lot. For instance, there are these different schools. For instance, one saying that the virus-like organisms were the first ones existing, and other schools saying that they could not exist before the cells were available because they are depending on the host cell. But then, in my mind, this is a question, but in my mind, something had to exist before the cells. So, and it had to be something far more simple than the cells. So it had to be, well, viruses are in the same range of simplicity at least. And as we have been saying, that most likely this very simple thing had to be confined in something which made of simple cells, although biological cells did not exist. So, actually, something? Yeah. But I think, something like viruses have been existing before cellular hosts.
But there are no viruses known nowadays.
We can certainly make viruses replicate in test tube. Just give it the right things. You can mimic the cell, like maybe the early world just mimic the cells.
How large a fraction, or how much of a virus can you produce in your lab?
In the lab?
Well, I mean,
you mean
is it half of a virus or a tenth of a virus?
Well, people have cloned many viruses full length, but they, of course, that means that they exist in the nation. People just copy them and they do test tube. So we can maintain viruses and manipulate them, in cloned form, and do any kind of manipulations to the clones and then convert them back to viruses. But make from scratch, I think that would be a very interesting question, and nobody can make it from scratch. Somebody.
Do you have a feeling for how much? What the fraction of a virus can you make from scratch?
1,000.
Well, you can build it, but you don't know if it functions.
The leg or the head or whatever.
You can synthesize the whole thing. Of course, as soon as we know the sequence, we can make it, but we have to know the sequence first. So if we would try to guess a complete sequence out of blue, that would be difficult. But certainly we can just assemble it if we...
We can make any sequence.
We can make any sequence. We can... But synthetic cells is again more difficult. We can make these non-cellular organisms, and we can generate life to that level, but we cannot make the cell.
All right then. So thanks very much, Kirsi, for this lecture. There's so much biology. So now we have.